Hello everyone, my name is Caitlin Wolf and I am a senior working on this research project under biology faculty member Jeremy Weiss. During our time this year, we were also aided by junior Grace Ryman in completing this work, so a big thanks to her for what she was able to accomplish. Our research is focused on the microbiome of Oregon wine grapes and how environmental variations and farming practices influence the microbes present. To begin, a microbiome is defined as a group of microbes which include bacteria, fungi, and viruses that are found in or on an organism. Microbiomes are ever present in the world around us as well as within our own systems. Some particularly famous examples of these are the human gut, which aids in the digestion of food, thermal ocean vents, which acts as a primary food source for deep ocean environments, and plants. Viterus vinifera, or the common grapevine, is colonized by various species of microbes on each of its different tissue types. An example of the species that are common to specific locations can be seen in the picture on the right. The presence of these species, as well as many others, have been hypothesized to be affected by environmental conditions as well as how the vines are grown in a vineyard. Microbiomes are also exceedingly important for the winemaking process. The microbes present not only aid in the fermentation and metabolic breakdown of grapes for wine, but may also change the terroir or taste of wines made from the Oregon grapes. A study done in California helped answer our question if microbes do actually change based on location. The research tested the microbes of wine grapes in 3 different locations and showed that geographic location did in fact impact what species resided in the vineyards, as can be seen in the graph on the left. Between Napa Valley, Central Coast, and Sonoma, the species present in the vineyard clustered based on the location and therefore the environmental impacts that were present. The purpose of our research is to expand on the understanding of Oregon-based vineyard microbiomes since there is currently very little data presented. And since Oregon wines are so recognizable worldwide, could the microbes present be what sets them apart? In order to test this in Oregon, characteristics of vineyards were identified: geographic location and farming practices. To identify location, vineyards are nationally separated by the American viticulture areas, also known as AVAs. In Oregon, there are 18 distinct AVAs, 5 of which were used during this project and are listed in the picture below. To distinguish farming practices, vineyards that were certified as Demeter vineyards were classified as biodynamic, while those that weren't were considered conventional. The Demeter certification is internationally recognized and certifies that a farm of any sort practices an organic-style farming where no GMOs or herbicides are used. This is contrasted by conventional farmers where vegetation is controlled by herbicides to maintain vine health. With that being said, the majority of our conventional vineyards still did use eco-friendly herbicides in order to make as little environmental impact as possible. Here is a list of the 10 vineyards sampled during the 2018 vintage. 2 vineyards under different farming classifications were sampled from each AVA to allow the comparison of both variables of interest. Unfortunately, samples taken from the Brook Vineyard did not yield reliable bacterial data, so the bacteria results were removed from the group, although fungal information was still used in the analysis. From the information we have gathered, the hypothesis was drawn that vineyards under different farming practices and AVAs will have less similar bacterial and fungal communities than those that share these characteristics. The methods of this research began by identifying what vineyards were going to be used based on location farming. In the field, 5 Pinot Noir grape clusters were taken from a single block at each vineyard. 1 sample was collected every other row from the vines on the north side that were 10 paces in from the right side of the block. Back in lab, 4 random berries from different places in the cluster were removed and mashed to create a homogeneous liquid sample. Using the FastDNA spin kit for soil, DNA was isolated from the liquid and sent to Oregon State University for sequencing. The primers used selected specifically for bacterial and fungal 16S genes in order to identify the species present, but some off-target sequences were identified as well. The most common of these were bacterial DNA followed by mitochondrial, but since these organelles are hypothesized to have evolved from bacteria that were engulfed in pre-eukaryotic cells, it is understandable that these sequences were present. Once we received the sequences from OSU and removed any species that were not bacterial or fungal, the data was analyzed using RStudio to compare the species present in each of the sample to their assigned vineyard characteristics. The pictures shown on the left are a fun sneak peek into the fieldwork we did in 2019. We were very lucky to be able to sample in some beautiful areas last year. From this analysis, non-metric multidimensional scaling, or NMDS plots, were made. To create these plots, RStudio made a multidimensional graph to plot each of the sample data points based on the variables we were asking it to consider. Once plotted, the system statistically alters a plot back to a two-dimensional space in order to show the clusters at a more understandable level. To read these at a basic level, polygons that are overlapping are more similar to the polygons that don't, although more statistical analysis must be done to confirm this. The numbers on the x and y axis are completely meaningless for the work that we are trying to do here, so they can be disregarded. On the left, the bacterial communities in the two farming practices are compared and shown to have significant difference between the two. On the right, we see that the fungal communities are compared and show to be very similar despite being under different agricultural methods. This data suggests that the bacterial communities are affected by the farming practices. Here, we compare each AVA. On the left, we again have bacterial communities present, and we see that there is no difference between the AVAs with the exception of Anola amity. On the right, the fungal communities are shown to be significantly different from one another. This suggests that the fungi present in the vineyards are more affected by geographic location. While the NMDS plots show the overall presence of species in the sample, diversity of the samples were also considered here by using the Shannon Index of Diversity. This index considers diversity and the evenness of species abundance in each vineyard. The diversity between farming practices for both bacterial and fungal communities were insignificant, so only the diversity by AVAs is shown here. To read these plots, bars that are labeled with different letters show significant difference compared to those that share letters. On the left, diversity of bacteria shows that there is a variable significance throughout the locations, with Anola amity again being an outlier. On the right, fungal communities diversity shows a similar result, suggesting that the microbiome diversity levels are highly variable and highly affected by location of the vineyard. From the data gathered, we were able to support our hypothesis that microbiomes vary based on location and farming practices, further suggesting that these changes make terroir variable as well. An interesting discovery made was 6 strains specific to biodynamic vineyards and 5 to conventional, showing that microbes are greatly affected by farming practices. In 2019, we were able to partner with the University of Oregon graduate students to continue this work, allowing us to expand AVAs and grapevine tissues tested. We gained 2 AVAs from the Columbia River Gorge and began sampling Pinot Noir tissues, including grapes, soil, bark, leaves, and wine samples throughout the fermentation process. By increasing the tissue types tested, we were able to observe a more complete view of grapevine microbiomes and see how communities change during fermentation. Unfortunately, due to our world's current circumstances, the data gathered during this time was unable to be completed. This work will continue, however, in the following year and hopes to further isolate microbiomes of Oregon vineyards and begin to test for correlations between terroir of wines and present microbes. To finish this presentation, we wanted to take the time to thank those that helped make this research possible. First, we'd like to thank Alexandra Morris, class of 19, and Shay Gerscher, class of 18, for their work towards understanding vineyard microbiomes in previous years. Second, we would like to thank the Oregon Wine Board for the grant that funded this project, and specifically JW and Greg Jones from the board who helped make it happen. Lastly, I would like to thank you for attending this virtual presentation and learning about the microbes present on Oregon wine grapes. We hope you have enjoyed this presentation and continue to stay safe. Thank you.